Hey, welcome to Notes, and today we are going to see how to interpret a phylogram. Okay, we have a series of lectures to show you how to draw a phylogram using blast analysis, reference sequences, multiple sequence alignment. Using Mega to align again and draw this phylogram. Okay, so before we proceed, I want to highlight few things. There is a distinct difference between phylogram and cladogram. For example, what are you seeing here is a cladogram. How to differentiate them and what is cladogram? Cladogram just tell us the grouping patterns among the DNA sequences. Okay. For example, see here, the branch tip of this cladogram is all uniform. Okay. So definitely they are not the phylogram. Phylogram will tell you the similarities and variations of DNA nucleotides or nucleotides among the various species. Also, if I see some most of the times in the Mega, this option is selected by default. You can just unselect this option. You will have a phylogram. See the branch tip now. They are all uneven. How? Why? This is something called tree scale. You will see only in phylogram, not in the cladogram. Cladogram will not have a tree scale. Only phylogram will have a tree scale. So what is this number? Numerical value represents. This length in the tree scale represents the zero point zero 0.01 nucleotide substitution between the sequences. For example, consider this sequence Pseudomonas aeruginosa with Pseudomonas tenensis. When you see it is almost two times equal, two times lengthier, the Pseudomonas tenensis. Okay, what this means is they may have harbored 0.02% of nucleotide differences between Pseudomonas aeruginosa and Pseudomonas tenensis. Okay, so this length corresponds to this many nucleotide substitution between the sequences. That's why we have a tree scale. Okay, say for example, in this study, this is my target sequence, the sequence I have generated in this study, and they did not even have a point one, simple branching tip, meaning these two sequences share exactly 100% similarity. All nucleotides in this sequence, they found a match with this sequence also. So they don't have any variation. They are 100% similar. So this tree scale is an yeah, measuring scale of number of nucleotide substitution between the two DNA sequence. And these are bootstraps. When you see their reliability of branching pattern, when you see 100% bootstrap, they are definitely two different branches. When anything less than because it is a reliability of branching pattern, you are finding them in the nodes of every branches. Anything less than 70 is not a reliable branching pattern. For example, here. Okay. So apart from this, you will have something called captions. Sometimes they are, hidden like this. They are not visible. Sometimes you will see them here, or you can select this option, caption, copy to clipboard. You can, have this option selected. Okay. And I'm also finding many people are taking screenshots and, sharing the script pictures in the manuscript. Not necessary. You have an image option, PNG option. You can click it, save it anywhere. Always use a separate file because many times during your manuscript communication, you need to have a file separately for communication. Okay. So these are the various options here. So how to interpret the phylogram or why? Phylograms are for studying phylogenetic signals, but because there is a cladogram option and also phylogram option, you can also complement your blast result with phylogenetic analysis. For example, definitely the sequence I produced in this study is Pseudomonas aeruginosa because I sequenced 16S rRNA gene and it is cladding exactly with the Pseudomonas aeruginosa clad. Okay, if you see the phylogram from here, there are two types of branches. Okay, type say maybe branch A, branch B. Branch A again have two sub branches, one with nicosulfurodens and other with citronellosis of Pseudomonas. And another branch exclusively have Pseudomonas aeruginosa. And my sequence cladded perfectly without any tree skull variation. So definitely this is Pseudomonas aeruginosa. And there is one more information in this tree. If you see this nicosulfurodens and the citronellosis, these are all pathogen. They have reported in many manuscripts as an opportunistic pathogen or hospital acquired infections. But these species of Pseudomonas that has been cladded here in the topmost branch, all are environmental isolates. If you see check the publications, you will see these are all environmental isolates. So it is very interesting to see how attribute of Pseudomonas species are actually reflected in the phylogram. Pathogenic attribute and environmental attribute. That's how these bacteria are divided. Likewise, we can use phylogram for various other interpretations. Okay. So if you have any queries, you can post it in the comment box. That's how we produce, publishable quality of phylograms. Thanks for joining.